Lokesh Guglani, MD, who's an associate professor in the PACS division. He received his MD at the University of Delhi and then residency at SUNY Buffalo and fellowship at Children's Hospital of Pittsburgh before he joined the faculty in the Department of Pediatrics at Wayne State University in Detroit. Then we were able to recruit him very happily to Emory Children's in 2015, where he's really one of the most creative physician scientists I've had the pleasure of working with. In addition to being one of the key providers in the CF program, Dr. Guglani also contributes to the pulmonary hypertension and vent-dependent clinics, and has recently established the new primary ciliary dyskinesia center. Lokesh is going to then hand off the baton to Rabin Tirouvanziam, PhD. Rabin is also an associate professor in the PACS division. He received his training as an engineer and his PhD in immunology and developmental biology at Agro Paris Tech in France, and then postdoctoral training at Stanford University. He became an instructor at Stanford for 4 years before we recruited him to Emory Children's in 2011. In fact, he was the first recruit to our new CF Research Center. Rabin has since gone on to become one of the key researchers in our center, leading a large and growing team, and has been very successful in obtaining extramural grants and turning those grants into important research advances. And today, Lokesh and Rabin will present their joint work on a really excellent project that grew from institutional support to now having extramural support and is continuing to grow. And I really love this project because it's a fantastic example of a partnership between a basic and translational scientist and a physician scientist to solve really important problems in CF disease. And so we're going to hear from Lokesh and then Rabin. And I've asked Dr. Rachel Linneman, who's the director of our CF clinical program, to handle the Q&A session at the end. So Lokesh, take it away.
Great, thank you so much. And thank you for this opportunity for presenting our joint efforts. So I'll be talking about this program that Rabin and I have started, which is titled Integrated Monitoring Platform for Early Disease Events in Cystic Fibrosis, hence the acronym IMPEDE-CF. So let me see, oops, moving too fast. So just to give you a little background about what has been happening in the CF world, what have been the major milestones that we have achieved in the last 10 or 15 years. Gone are the days when people would present with symptoms and we would make the diagnosis based on presence of symptoms. Newborn screening, in fact, has now become the most common mode of diagnosis of cystic fibrosis. And that has led to a significant improvement in immediate nutritional outcomes as well as long-term survival, because they're started on therapies much more promptly now, as opposed to waiting until symptoms develop. The other big game changer has been this recent introduction of CFTR modulator drugs that have completely changed the landscape of CF care because they go after the basic defect in cystic fibrosis. And this is illustrated nicely in this schematic here, where these different drug combinations have been shown to steadily improve the lung function in patients with cystic fibrosis with various combinations of mutations. As you can see that the initial generation compounds had much lower efficacy, but as the second and the third generation products have been released, we've been able to see much more significant improvement. The goal is to restore CFTR activity. So if people believe that if you can restore at least up to 25% of CFTR activity, you can reduce the clinical manifestations of the disease. And now you can see that these newer generation compounds are able to restore much more than that. And in fact, that sort of ties along with the incremental improvement in lung function as well. So with that, we look at how does lung disease begin with in the first place. We know that it starts off right away, even though children are mostly asymptomatic. And so this is the era, which is the silent phase of cystic fibrosis, where you have minimal symptoms, but you have small airways getting involved that then starts to spread to sublobar regions, which then incites this inflammatory response with influx of neutrophils and a whole lot of other changes that accompany that, including proteolysis caused by the neutrophil elastase that's being released. There's oxidative stress in the airways, there's local hypoxia, and the mucus overproduction tends to cause significant airways obstruction. So what starts off as epithelial dysfunction, then combined with the immune imbalance, leads to entry of microbes into the airways. And all of these factors combined eventually result in airway destruction, which leads to bronchiectasis, which is the end result of the disease. So we do see this progressive deterioration of lung function over time in our patients, despite all of our efforts and interventions. And what are we doing in clinic? We're basically looking at the tip of the iceberg, because we have very rudimentary tools still in this day and age to evaluate our patients and their lung disease. So we rely on simple things like chest x-ray or a clinical examination, and we're trying to avoid the development of bronchiectasis in our patients. But what is happening beneath the surface is not what's routinely monitored, and that's what we are hoping to change. So with this schematic, it's showing that there's mucus plugging, airway wall thickening, and development of trapped air, which are early changes of CF that can be evaluated on T scans. You can also look at airway fluids to look at their inflammatory markers and measure lung function by using more sensitive techniques such as the lung clearance index. So to summarize the changes that happen in early CF lung disease, clinical symptoms are somewhat minimal. We are looking for structural changes, which is bronchiectasis that can be seen on imaging, functional changes that could be measured by lung function testing, but it's the biological changes that have been sort of not very clear, been very clear in terms of which comes first. Is it the inflammation? Is it the infection? Is it the obstruction? And what is the sequence or the interaction between these three that leads to the progression of lung disease? And now you add into the mix this CFTR modulators that changes the natural history of the disease, that makes the situation even more complicated. So we have some data from earlier cohorts that have looked at this in terms of the effect of early CF lung disease. And the data mostly comes from the Australian group, which started this cohort of Arrest CF back in early 2000s, where they were able to achieve a lot of important developments. The most important being that establishing that early lung disease starts to appear, and by 3 years of age, 50 to 70% of patients have had bronchiectasis. They also developed this validated scoring system for evaluating lung disease on chest CT. And their seminal paper in the New England Journal showed that neutrophil elastase activity in BAL correlates with structural changes that we see on CT scan of patients at 3 years of age. However, this data was somewhat limited in that it was mostly qualitative assessment of neutrophil elastase, and there are much more sensitive techniques available now to assess neutrophil elastase level, both free and bound. And so we'll talk a little bit about that in the talk by Rabin. And all this has sort of led to this collaboration with these centers. So we now have a multi-center group which works collaboratively on these studies on early CF lung disease. So our questions were mainly, is it possible to monitor airway inflammation in children in a non-invasive manner? Can we develop specific biomarkers for early stages of lung disease? And can we identify specific targets or signaling pathways that are involved that could be useful in developing interventions for the early stages of lung disease? And then also finally understanding better, what is the effect of CFTR modulator therapies on this inflammation in the early stages of lung disease? So that's what led to the creation of this program, which is a prospective longitudinal cohort study where we basically are looking at non-invasive sampling of the airways sequentially in patients who are enrolled during infancy and then followed annually and during their acute illnesses. So we enroll patients between 3 months to 6 years of age with a clinical diagnosis of CF that are being followed at our center. We also have control subjects that are undergoing bronchoscopy for other non-CF inflammatory airway diseases. And this also includes patients from the aerodigestive clinic, patients who have primary ciliary dyskinesia, as they act as an important control for cystic fibrosis as they end up having bronchiectasis as well. So we excluded patients for any significant prematurity, if they had significant airway interventions, if they were tracheostomy dependent, or they were on cytotoxic medications. The visits were comprised of the enrollment visit, followed by the annual visit every year when they would get their annual labs drawn, and we would collect some additional samples at the same time. And then, as per the CF Foundation mandated imaging protocol for early CF lung disease, where CT scans are done at ages 2, 4, and 6 years, we would coordinate with those visits and try to perform a bronchoscopy at the same time as the CT scan, because most 2-year-olds would require sedation for their CT scan. So we use that opportunity to be able to do a more thorough assessment of their lower airways, and we'll talk more about how that's done and why it's important. And then additionally, if they were to be hospitalized, we would collect additional samples as well. So bronchoscopy remains the gold standard for the assessment of lower airway infection and inflammation. And in our case, I was lucky to be working with a great team of people at Children's Healthcare of Atlanta in facilitating this happening, because it involved coordination with radiology, anesthesia, operating room, and the parents to try to make it happen all on the same day. So we collected induced sputum, exhaled breath condensate, a blood sample, and performed the chest CT and bronchoscopy all on the same day within the span of 3 to 4 hours on a given morning. And most patients would go home by about noon of the same day. What we did was that we also understand that this is an invasive procedure and that is being done in a vulnerable population. So we actually set up a data safety monitoring board to monitor the invasive procedures that are part of this study. And this was constantly, the data safety monitoring board was constantly updated with quarterly progress reports, and they reviewed and provided their recommendations on a quarterly basis as well. And I'm happy to report that we did not have any adverse events. The next technique that we used is induced sputum, which is a standardized technique where we basically collect some secretions that are brought up with the help of a hypertonic saline nebulization, along with the chest vest to shake those secretions loose. And this is safe to perform in infants and young children. It's less invasive, and it can provide a lower airway sample, but you have to be careful with its interpretation, as we will discuss further. But it does provide a good sample for the assessment of both inflammatory cells and infectious status. And this was based on the protocol that was developed in Europe by the European Cystic Fibrosis Society Clinical Trials Network. So we adapted that for our use here in the United States. Chest CT was our other major method of assessment of lung disease. It remains the gold standard for assessment of structural changes. And what we created in collaboration with our colleagues in Netherlands was this standardized protocol, which collected both inspiratory and expiratory breath hold scans. And why is that important? Because you can miss a lot of changes if you just look at inspiratory films. Shown here in this schematic is, on the left, an inspiratory scan at a certain level in a patient, and the same scan in the expiratory phase. And you can see these areas of hypoattenuation that are representing areas of air trapping that are clearly seen much better on an expiratory film than they show on an inspiratory film. So we can detect small airways dysfunction in a much more sensitive manner by looking at expiratory images showing these areas of trapped air. And once we have these CT scans, we are able to objectively quantify the disease, because in early CF lung disease, the disease is quite regional. So we understand that just by looking at a CT scan, you may not be able to see all of it. And so this technique that was developed, what it does is basically overlays a grid of squares on each slice of the CT scan. And then you're able to quantify the disease in each square. And as a result of this quantification, you can calculate a total percentage of how much lung is healthy and how much is diseased. So shown on the left here is an inspiratory scan where you can see several areas that show airway wall thickening, airway dilatation suggestive of bronchiectasis. There are areas of mucus plugging seen in yellow. The small pink areas are the areas of atelectasis. So you can quantify all different aspects of lung disease in a very specific manner, and this can be tracked over time. Similarly, in the panel on the right, we're seeing an expiratory scan where the areas in blue are marking out the areas of air trapping. So you can quantify the degree of air trapping quite nicely. So this is in keeping with the preschool guidelines from the Cystic Fibrosis Foundation, where they have established the routine use of CT scans, and this is what we have been following at our center as well. So all this data that's being collected as part of this study includes clinical data, imaging data. There's a lot of cellular and biochemical data that Rabin's going to talk about that is processed by his lab, and then metabolomics is done by Joshua Chandler, whose lab does both targeted and untargeted metabolomics. And all that data is integrated, and with the help of our biostatistician, Limin Peng, who's at the Rollins School of Public Health, we're able to conduct data analysis. So let's talk a little bit about what we've done so far. We've been able to, in about less than 2 years, enroll about 49 patients. And of those, about 11 of them have had this protocol where they had the CT, bronchoscopy, induced sputum, exhale breath collected all on the same day, which in 5 of those actually were done before they were started on this CFTR modulator therapy. So we're really interested in seeing what happens to these patients after they have been on this therapy on subsequent follow-up. And as I said earlier, we did not have any adverse events or hospitalizations from any of the study-related procedures. So this is just a demographic table showing the data for those 11 patients that we have the complete workup for. As you can see, that Delta F508 tends to be one of the most common mutations. Most of these patients are infected with relatively benign organisms, and these are the patients that had or can be started. Most of them were pancreatic insufficient, not a whole lot of hospitalizations for pulmonary exacerbations, and their nutritional status overall was pretty good. So these are the CT data. So this is showing percent disease. I do want you to pay attention to the y-axis, which represents the percent of total disease in the lungs. Now remember that these are 2-year-old subjects, so you don't want them to have too much lung disease to begin with. And so what is incredible is that even at this age, for these relatively so-called clinically stable patients, we do see up to 6% of their lungs involved with significant disease, although there's variation across the spectrum depending on mutation type and disease severity. And then if you look at bronchiectasis, which would be nice to be as low as possible, but still we're seeing up to 1% of bronchiectasis starting to appear in some of our patients here. And then trapped air was pretty low for the most part, except for this one outlier. So that's also helpful to track as well. Coming to induced sputum collection. Now, this is something that obviously was done in the pre-COVID-19 era. So we were able to do this, but we can certainly talk more about what should we be doing in this day and age. But so far, we've done 23 procedures. There was a bit of a learning curve to this because we had some initial failures, most of it because there were some non-cooperative subjects, or maybe we just didn't get enough sample on a given day because these patients were coming in a fasting state for their procedure in the morning, and so their mouths were pretty dry and they were just not, very in a good mood, I should say. But nevertheless, we still got a pretty good cell yield from these samples, and the volume that we got from these subjects, even in an acute pulmonary exacerbation, was fairly usable in the lab. And then the other technique we used is exhale breath condensate, which is a simple technique which basically condenses the water as it's coming out in your exhaled breath. And then you collect that in this long tube that's covered by this steel casing that is frozen and basically allows the water vapor to condense down in the plastic tube inside and allows you to collect that to be analyzed by mass spectrometry. We had to modify the setup quite a bit because of our population being so young. So we actually took away the mouthpiece and we actually attached a face mask to it just to make it more convenient, because 2-year-olds cannot put their lips around a mouthpiece and maintain an adequate seal for 10 minutes. So we had to come up with these modifications and also add some inhalation filters and exhalation check valves to make sure it works without causing any significant environmental contamination of the sample. So this is what we found in terms of our yields for the R-tube, for the exhaled breath condensate collection, where we found that the regular setup, which is the standard, what is used in adults, did not work for us. We had very low yields initially, but then once we modified our setup, we had significant improvement in our exhaled breath condensate yields. We also actually tried using the R-tube vent, which is a modified version that we attached to the anesthesia circuit as these patients were undergoing bronchoscopy, but we found that there was a lot of contamination from the anesthesia gases that were collecting inside as well, so we kind of stopped doing that. But overall, we had a pretty good collection from these tubes with a very low failure rate. So with that, I would like to hand off to Rabin, who's gonna present the basic science data that comes from the PCF, and I'll be happy to answer questions at the end.
Thank you, Lokesh. So hopefully you guys have my PowerPoint now. That's showing up. Do you see that in presenter mode, or do you see that with a regular slide?
It's a regular slide.
Regular slide. Oh, okay, excellent. So, well, thank you, Lokesh, for the beautiful setup for me. Obviously, these types of research, as Niall was pointing out, are only possible in tight collaboration between clinicians and basic scientists like myself. So, huge kudos to Lokesh for bearing with us as we are constantly hammering him with requests for this sample and that sample. And I think it's been an extremely productive collaboration in the last few years we've been working together. So our process is fairly simple. What we're trying to do is we're trying to do as much as we can with the samples from patients that we get. And clearly in CF, the disease starts in the airways, while systemically, kids are doing fairly well. So we're using whole blood really as a compartmental control for what's happening in the lung. And as Lokesh mentioned, bronchoalveolar lavage is really the gold standard for collecting samples from these children, but it comes with a lot of issues, obviously invasiveness, the fact that the kids need to be sedated. So we've been really interested in trying to see if we can find less invasive alternatives like induced sputum. We have developed over the years a method whereby we don't use chemicals. We just use simple saline method to dissociate the airway samples from CF patients. This has been working for us quite beautifully. We don't really modify any of the metabolites, or especially the redox equilibrium that we're really interested in. We're able to quickly homogenize those samples and centrifuge them, and then we generate several types of fractions. And downstream of those fractions, we do a number of assays to try to understand what the cells are telling us, also what the contents of the fluid are in terms of immunomodulators, cytokines, enzymes that might be released by epithelial cells or the immune cells. And we also look obviously at the microbiology. The obvious, I would say, difficulty with the samples from babies is obviously the low volume. And also the fact that the disease is just starting. So for example, what I'm showing down here is that neutrophil elastase, which has been over the years a staple of monitoring of disease in CF patients. It's an enzyme coming from neutrophils, and it chews up the extracellular matrix, so it's directly related to the advent of bronchiectasis. This has been, again, something that's been measured very efficiently in adults or children. As you track back to the very early stage, we find that elastase activity is actually very, very low, even with the best methods we have. And in fact, in this particular study, in PCF, we found now that in the induced sputum, we have very low levels of elastase. So we cannot count on elastase to tell us what's happening in the lungs. So we have to look at other biomarkers. So let's go through them. One category, an obvious one, is cytokines. So the cytokines are those mediators that are generated by host cells to regulate and coordinate responses. So they can be generated by the CF epithelium. upon stress. They can be generated by immune cells that are either resident, like the macrophages, or immune cells that might be recruited into the lung, like the neutrophils or the inflammatory monocytes or T cells. And our game as immunologists is really to look at these different cytokines, and based on the signatures that we find, be able to reconstruct, is this coming from epithelial cells? Is it coming from neutrophils, T cells, et cetera, et cetera. So IS is a completely new matrix for basic scientists to work with in early CF. So really, the last couple of years have been spent trying to validate the use of induced sputum and compare it to the gold standard, which is BAL. So what I'm showing here is a cross-correlation between bronchial lavage and induced sputum that were collected at exactly the same visit on our children. And we're looking at a number of different molecules here. No need for you to just go through the list and figure out what they are, but by and large, what we see is that there's a group of let's say half a dozen, which seem to be very highly correlated between BAL and IS, and a bunch of others that really don't show much of a correlation. So this really means for us that we're sampling different areas of the lung, which we knew already, but we had to prove it. And also that some of the cytokines that are typically measured in BAL will not be reliably measured in IS, but vice versa. Some of the things we can measure in IS will not be well measured in BAL. So I'm showing here a few of them, which are actually easily measurable in both matrices and are clearly different between those airway matrices and what we find in the plasma in red. And we're showing some statistics here in paired manner. So we're really pairing the systemic compartment to the different airway matrices. So we find, for example, for the VEGF and for IL-8, which are mediators that are linked to neutrophilic inflammation, there is readily in this early cohort of 2-year-olds an increase that we can find both with BAL and IS compared to plasma. And that holds also for molecules like MCSF, which is a colony-stimulating factor for monocytes and macrophages, or GCSF, which is a colony-stimulating factor for granulocytes. So even though these children don't really have a lot happening at the structural level, they already have inflammation based on these immunomodulators. Now, this is a sort of a complicated graph. I'm going to take you through it. It's actually fairly simple. We have BAL on the left-hand side, induced sputum on the right-hand side. What we did here was to calculate cross correlations between the top 3 outcomes, which are derived from Lokesh's work on the CT scan. So percent disease, and then there's percent abnormal airways. And basically what we look at here is whether all the other outcomes here, which are cytokines measured either in the bronchial lavage or in the IS, have any correlation with one another, but also have any correlation with the pragma scoring, the measure of structural damage by CT scan. And you can see here, you can eyeball it. It's very simple. You see the first 3 columns, and there's basically no correlation between the structural damage and the cytokines. So we're at a stage where, again, disease is very is starting, and structural damage is really not very substantial yet, even though we can measure it. We can measure the cytokines as well, but unlike what we see later on in the disease, those cytokines really do not track with the damage based on CT. So what are our other options? Well, one of the things we figured out a while ago, and this was a product of our collaboration with colleagues in the Netherlands at Erasmus University in Rotterdam and colleagues in Australia, in Perth and Melbourne. So we've pulled together our resources and looked at bronchial velar lavage over more than 50 or so patients. And we used the method that I developed when I was back at Stanford as a postdoc and brought back in my lab here at Emory, a method whereby we can actually collect the cells from the airway fluid. So we're not looking at the components of the fluid itself now, we're looking at the cells that are contained within this fluid. And instead of just doing cytospin and looking at, okay, is it a neutrophil? Do we have T cells? Do we have macrophages? We use flow cytometry to do single cell analysis of the actual activation state of these cells. And one of the things we figured out very early on when we were looking at adults and then tracking back at earlier and earlier stages of the disease is that in cystic fibrosis, the epithelium is calling for neutrophils from the circulation to come and migrate into the lungs. And those neutrophils are releasing mediators as we know it. But we discovered by our deep analysis of these cells in adults that the reason or the way by which these neutrophils release those toxic molecules is by a completely different mode. Instead of bursting out and dying, which was what people were thinking, they're actually actively responding to that microenvironment, and they're taking some of their granules that are pre-made in the bone marrow, and they're putting them to the plasma membrane, and then they're releasing those contents into the lumen. And that's the reason why we have, for example, elastase showing up in the fluid of CF patients, is because the neutrophils are being told by this microenvironment to actively take their elastase from the granules and put that elastase outside. So we have a way to actually measure that process by flow cytometry. We can use a molecule that's at the surface of these intracellular granules, and when those granules are put to the surface of the neutrophil, those molecules now show up at the membrane and the plasma membrane of the cells. We can bind antibodies to it, and we can measure that by fluorescence. This is what we call the CD63. So this is that molecule that we measure as a median fluorescence intensity. As you can see here, in that large cohort that we studied a couple of years ago, when we plot the rate of active release of those mediators by the neutrophils in the airways against the CT scan measure of lung damage, we have a very, very strong correlation. So we basically show, even though elastase might not be measurable at as a molecule yet, because it has some inhibitors and other things that make it difficult to measure at that early stage, we can catch the cells in the process of releasing this elastase, and this is a very stable measurement that we can use. So we've published that in the Blue Journal a year ago, and obviously our game in this IMPEDE-CF study was to see if we could find the same thing in the induced sputum. Nobody had tried to measure immune cells in the induced sputum of infants before. So the first thing we had to do was to show that by flow cytometry, we were able to pick up the different cell types that we're interested in. And here I show the type of analysis we do in the lab. This is what we call a gating strategy by flow cytometry. As a reference here, I'm showing you blood on the left-hand side. You can see that out of whole blood, we can easily measure neutrophils, eosinophils, monocytes, lymphocytes, and we can, go sort of downstream from these big populations. And from lymphocytes, for example, we can pick up, CD4, CD8 T cells and B cells and NK cells, et cetera, et cetera. Obviously, in CF, we're really interested in neutrophils and macrophages because they are the main cells that seem to be responsible for the beginning and then the progression of the inflammatory syndrome. So what I show here in the middle and on the right-hand side is that in both bronchoalveolar lavage and induced sputum, we have very efficient ways to pick up live neutrophils that are there, as well as live macrophages. And you can see that those populations show up in the induced sputum very robustly. So now the question was, can we measure exocytosis or release of granules in the neutrophils in the induced sputum? And does that track with lung disease, as we have shown previously with bronchoalveolar lavage? And the answer is yes. So, and this is actually, for us, a very, very significant result. It was really the basis of our multi-center study that got funded last year for the CF Foundation. So I should say, within a year, we've already sort of confirmed our main hypothesis in this 3-year project. So showing you here in a little bit more detail, when we compare blood, bronchoalveolar lavage, and induced sputum from these children, we can very efficiently measure monocytes, macrophages, and T cells. And you can see that at that stage, because inflammation hasn't really picked up, or gained a lot of steam yet, the microenvironment is still dominated by macrophages. So this is sort of the normal picture you would see normally in bronchoalveolar lavage, 70 to 90% macrophages. And in IS, it's even more the case. You really have a dominance at that early stage of those macrophages. T cells are at very, very low levels in CF patients at that stage. The neutrophils are there between 10 and 20, 30, 40% maybe maximum. But the key here is this measurement here on the bottom, the percent of those what we call GRIM neutrophils. So these are the granule releasing and immunomodulatory and metabolically active neutrophils. This is a signature of disease that we discovered. And we find that this signature is readily present in the induced sputum. And now we'll show you a cross correlation similar to what I showed you earlier with the cytokines. The disease score that Lokesh measures on the CT, so that's a purely clinical measurement. And now we get the cells from the bronchoalveolar lavage and we get the cells from the IS. We get also the fluid. And here I'm basically illustrating for you that if we measure those exocytosing neutrophils in the BAL, there's a correlation with the pragma score. If we measure those exocytosing neutrophils in the induced sputum, there's a very strong correlation with the pragma score as well. And the correlation exists also between those two compartments, okay? What's interesting at that stage, as we showed before, is that the measurement of elastase itself, the byproduct of that release process, that activity is not yet, has not yet overcome the anti-protease shield that's in the airways. So it's not really emerged as yet as an efficient sort of biomarker that you can track and that allows you to basically sort of reflect what's happening on the the structural level. So this is just to show that the cellular outcome at that stage is superior to the fluid outcome. Okay, I'm going to move on a little bit, and I'm going to give credit to sort of the third musketeer that we have in our team on the lab side, Joshua Chandler. I'm going to show a picture of him. He's definitely better looking than myself, and I won't say anything about Lokesh because he's a good-looking guy. But Josh is really an expert in metabonomics, and we've been working together to try to find if we can derive from the fluid, instead of enzymes, maybe metabolic byproducts of those enzymes that would be more stable and that we could measure in the different matrices that we get from those airway matrices that we get from those patients. And again, this is a paper that we published with Joshua and Lokesh and our colleagues in Australia and Netherlands a couple of years ago, where we took basically the hypothesis that as neutrophils release those special granules, what we call the primary granules or the elastase-rich granules, at the same time as they release elastase, they release an enzyme called myeloperoxidase. And those of you guys who might remember that from Immunology 101, myeloperoxidase is the enzyme that is present in neutrophils and monocytes, macrophages in our body. And these are the only cells that have this enzyme. Myeloperoxidase is the enzyme that is able to make bleach, okay? So this makes these cells highly potent at killing pathogens when they activate this pathway. And when the neutrophils get activated in CF airways, they release elastase, but at the same time, they release myeloperoxidase outside. So the hypothesis is when the release happens of those granules, myeloperoxidase should be present in the extracellular fluid, and then we should find byproducts of myeloperoxidase, including hypochlorous acid. What's important with hypochlorous acid is that it's going to oxidize a lot of targets, including methionine. And methionine, in that case, upon oxidation by myeloperoxidase, turns into that byproduct called methionine sulfoxide. And this is where it's great to have chemists and redox biologists like Joshua in our team, because when we handed over the bronchoalveolar lavage fluid to Joshua a few years ago, and he was able to measure a whole bunch of metabolites. What he did in this clustering analysis, right, is on the top, you can see the patients that are ordered by increasing structural lung damage. And what you see here in that sort of colorful palette is you can see a bunch of metabolites that are either low in children with sort of on the lower end of the structural damage measurement, and then start to increase as they accumulate more structural damage, or some metabolites here, those three in particular that are showing the opposite pattern. They're actually fairly high when disease is very, very mild, and they start to increase, they're high, sorry, when the disease is mild, and they start to decrease when the disease starts to worsen. And so there's a lot of different metabolites, all of them have important pathways that are linked to them. But I want to point out, we measured methionine sulfoxide here, and methionine sulfoxide, Joshua went on to show, is a very good correlate with increasing CT score. I want to point out also that the boundary between those different samples in terms of their metabolite profile, if you just look at the CT scan data, is very, very difficult to tell, right? You're basically within the range of 2 to 3% of pragma disease. This is where we think an inflection point occurs in the disease, and sort of the epithelium starts to empower the neutrophils to start to take, meaningful decisions into the microenvironment, and we know where that leads. So being able to measure that inflection point at such an early stage, where in fact by CT scan you start to see something, but you don't necessarily know whether the patients have pivoted towards the progressive phase of the disease. This is really what our joint program is about. So can we do the same in IS now that we've published it in BAL? So again, unpublished data from Joshua. He's preparing a paper. We're looking at it together now with Lokesh. Suffice it to say that Joshua has been able to show very broad metabolite coverage in the induced sputum, sort of similar to what he's been able to see in BAL. And we firmly believe that with some of the metabolites we find in there, we have the opportunity to survey the changes in the biology that are occurring at the cusp between these non-progressive and progressive phases, sort of similarly to what we've done with BAL. And I also want to give credit to sort of the exploratory arm of our joint study, where Lokesh has been showing a little bit earlier, coming up with a better way to collect exhaled breath condensate from our cohort of children. And then Joshua has been taking those 100 or 150 microliters of condensate and running that through his mass spectrometer. And here, really the kicker for you to consider is we're actually able to measure methionine sulfoxide, that byproduct of neutrophilic activation, release of myeloperoxidase, redox or oxidative activity. That signature is measured in the air that's being exhaled by our children, okay? And we can very, very sensitively and specifically measure it in the exhaled breath. So that's an area that obviously we're very excited in, and Joshua is leading that effort. So I'm gonna just switch now to sort of the end of our presentation, so that we give a little bit of time for you guys to ask questions. But I want to leave you with our working model. In the insert here, really what I want to highlight is we've discovered over the years that the neutrophils that are supposed to be very simple cells, all of us consider them as sort of pre-programmed, and they're mostly involved in bacterial phagocytosis. Well, in the last decade, people have shown that these neutrophils are actually pretty plastic. And in fact, they really decide what to do upon entering into a microenvironment. And they are being impacted by what the tissue tells them to do, by the presence of certain pathogens, be they fungi or bacteria or viruses, but they are also sort of impacted by what the previous wave of neutrophils that's already there tells them to do. So there's a lot of population behaviors that we have to keep understanding. I think a key basic discovery of our lab has been that in addition to this phagocytic fate of the neutrophils, and to the fate that's been discovered by some colleagues in Germany, whereby neutrophils are able to cast their DNA outside and form those so-called neutrophil extracellular traps made of chromatin, called nets. Our discovery has been that the neutrophils are able to stay alive in certain situations, but instead of endocytosing or taking up material and trying to kill it inside, they're actually able to cast their sort of active their effectors into the outside by very active degranulation, and this is the sort of grim phenotype that we call this granule releasing phenotype that for us now is really showing up as a very, very strong signature of CF, starting in our children. So the way we're looking at disease now is that really inflammation is starting very early. And I haven't showed you, for the lack of time, really all of our microbiology data. But at this point, with with the most powerful methods that we have, including metagenomics, we find that the airways of CF patients early on are not infected. They don't really contain any specific microbiota that we can differentiate from, that makes them different from normally developing children. Even though they really don't have a change either in the identity of those bacteria that are in the airways or in the burden, they already have neutrophils in the airways. And this is generally concomitant with some slight changes in the composition of the mucus. So the hypothesis that's now sort of gaining ground in our field is that early CF is linked to early inflammation prior to the advent of bacterial infection, but may be caused by some sterile inflammation, some stress, abnormal stress response that's involving maybe hypoxia of portions of the epithelium and a little bit of mucus obstruction. And what we know now is that the nasty bacteria that we're used to thinking of when we think CF, Pseudomonas, Staph, Haemophilus influenzae, those bacteria are actually coming later on, while neutrophilic inflammation is already in place in the airways. And later on, you have that stage where those bacteria turn into resistance form, so-called biofilms, and the amount of neutrophils increases even further. So what we're trying to really figure out is this very, very early stage on the left-hand side and see if we can target the neutrophils or even target the epithelium in ways that will allow disease to maybe stop at that stage where there's a little bit of something happening, but you really don't progress towards the full-blown inflammatory phase. So what have we learned so far? And I'm using the beautiful slides from Lokesh here. We know now that we can non-invasively monitor or monitor with less invasiveness the level of inflammation that's occurring in the airways of CF patients. We can do that with induced sputum, and we might have to answer some questions with you folks about the feasibility now in this new COVID era. But we can definitely also count on exhaled breath condensate, and this is really what we're continuing to push as part of this multi-center effort in the next 3 or 4 years. With regards to induced sputum itself, it obviously can be used to measure both fluid and cell-based biomarkers of inflammation, although we find that there are differences from what we're used to seeing in the gold standard bronchoalveolar lavage. For exhaled breath condensate itself, I think we need to look at modifications to be able to collect sufficient material for our studies. And obviously, we're excited that the Pragma CF method that was redeveloped by our colleagues abroad is now firmly in place with Lokesh. I think this method is very, very useful for us to figure out where patients stand in terms of their structural health of their lung. To finish with, really trying to hit the highlights of our homegrown program compared to our collaborators and other things that are happening in the United States and worldwide. We have really put the emphasis on developing new biochemical markers and trying to relate them very quickly to structural changes. So by focusing on some of these non-invasive techniques, we think that we have an edge, and we might be able to pave the way for studies that are going to be directed really at this, this, the inflammation that's occurring in the babies. Because of our consortium, we're able to very quickly exchange techniques, cross-train our collaborators, and ramp up some of these studies from our 50 or so patients in our cohort to 150 to 200 patients, counting all of our different partners in the consortium. We're talking every week with them. We're really cross-training both MD scientists and PhD scientists, and it's been an extremely a powerful tool for us to really provide new ways to look at disease in these kids. For us, the final objective is really to be able to look at what's happening at a very early stage and target this disease before it even starts to progress. So again, the IMPEDCF team, Lokesh, Lucas, his clinical coordinator, folks in my lab, in the Chandler lab, and then we have tremendous support from Li Peng, who's a professor in biostatistics, our collaborators in the Netherlands and in Australia. We also work with our colleagues at the Marsico Lung Institute at UNC, especially Chuck Esther, and also with Boucher, the head of the center. We started with the CF Air pilot grant. We're also very generously funded by the Hertz Family Foundation grant. And now we have solid support from the foundation, CF Foundation, for a multi-year grant. I'm going to leave you with a nice set of pictures from our collaborators, and then we're ready to take questions with Lokesh.
Thank you so much, Dr. Tirouvanziam and Dr. Guglani, for that wonderful talk. As Dr. McCarty mentioned, this is a prime example of scientists collaborating in the basic lab and the clinical spectrum. So I am happy to direct any questions to our presenters. If you have questions, feel free to send them through the chat. Maybe I'll start off with a question for Dr. Tirouvanziam, or for both of you really. What do you envision, you've taken the clinic to the lab, and as we think about translating these findings back from the lab to the clinic, what do you envision as the next step for neutrophil-based therapeutics?
So we are already developing with some of our Georgia Tech colleagues means, and we've published that recently in JCI with Krish Roy at Tech, ways by which we use that phenotype where neutrophils are specifically releasing certain proteases to target them directly in the lung. So we have some of these microgels that are lyophilizable, that can be nebulized into the lung. And when they swell, when they hit the lung surface, they swell and they release, they expose peptides that are protease sensitive on their surface. So we've engineered one that is actually elastase sensitive. So we find that we can specifically deliver into the lung, and only in areas where elastase has increased are those microgels going to be undone. And what we've done in this paper, it was published just 3 or 4 months ago, is we've actually delivered through those microgels that are elastase sensitive an inhibitor of elastase release. Okay, so in areas where neutrophils were activated, they were actually taking a drug that was making them unable to release the pathogenic factor. And with a single dose in our animal model, we were able to completely obliterate lung and systemic inflammation. So we think that there are real opportunities to target these cells once they are in the lung. Obviously, the other thing that we're dreaming about is, can we target these neutrophils while they are in blood on their way to the lung, and can we sort of change their poise such that when they come into the CF lung and they are exposed to certain cues from the microenvironment, they do not undergo this change and they do not release their elastase. Okay, we still want them in the lung, because if the epithelium calls them, it's because they have a job to do, and generally it's to help clean up some of the material that's accumulating in the lung. We just want them to do that and not necessarily start to create some damage there or start to recruit more of their kin.
Great, thank you. So a follow-up question to that from Dr. Jane is, can you speak to how this approach can form the basis of precision medicine in these patients? So perhaps also relevant to certain mutations or modulators.
I'll give this one to Lokesh, maybe.
Sure. So great question. I think this is, the way we would be proceeding in the future is to start to look closely at the mutation spectrum that we have in our group of patients here, and then to be able to come up with specific therapies that are introduced at specific time points based on the level of inflammation. Because as we all know that all of our patients behave differently because there are so many other environmental determinants of their disease. So I think knowing more about their mutation and how they're evolving over time will help us tailor those therapies according to their mutation severity. And in the future, I think that would help us tailor the introduction of therapies, because right now there's not a lot of consistency in terms of, let's say, simple things like hypertonic saline or pulmozyme introduction. So those could be perhaps adapted to specific mutation types.
If I may add just to that point, you have to realize that even though the gene CFTR is expressed in a lot of different organs, and we can track basically the severity of disease in certain organs to the type of mutations. So that's the case in the genital tract, or the case in the sweat glands, for example. We really don't have a good way to correlate severity of lung disease to residual function that's conferred by a given mutation of the CFTR. So clearly, lung disease is more than just not having CFTR. I think you can have, for example, the main mutation delta F508 can be homozygous for it and have literally no disease, or have very severe disease. Okay, so this is really where some of our measurements shine, because we need to go beyond simply the reading of the mutation. We need to actually determine within a single patient what their status is and how they progress. And I think this is critical. Structural disease is critical, but at very, very early stage, I think some of those biomarkers will be the only way by which we can figure out whether a preventative therapy will actually work.
Thank you. And we have a question from Dr. Joyner. What is the connection between dysfunction of CFTR and early inflammation in the lower airway, which is not a chloride secreting epithelium?
Good question. Lokesh, you want to take it, or you want me to take it?
Go ahead.
Okay. So, well, that's really the heart of the matter.
Yeah.
There are several hypotheses. I think one of them, still from our colleagues at UNC, who have been the proponents of the mucus hypothesis of CF, is that even though in the small airways you really don't have a sort of a complete layer of mucus that's covering the whole airways, and the mucociliary escalator really works in the upper airways, not so much in the lower airways, you still have patches of mucus here and there. And their hypothesis is that there's a little bit of upon stress or anything that can generate a little bit of hub in the epithelial cells. They will need CFTR to be active, maybe to flush some small areas. And if they're not able to do so because that CFTR is not acutely able to change the hydration of that layer, those areas get inspissated, and then the epithelium underneath basically dies of hypoxia. And this is the sort of the hypoxia hypothesis of inflammation. Okay, we've got things that are sort of similar but a little bit different. What we think is that what might create some of the issue in CF airways early on is, and it's interesting when we start to think about viral diseases as well, is that we know now that the epithelium and the macrophages are constantly releasing small vesicles that are viral-like to talk to each other, and those can constitute up to 1 billion per ml. So there's a lot of material that our lungs are constantly producing, those little particles. And we think that when those particles accumulate to high numbers, this can form a stress for the epithelium. So it doesn't necessarily have to be a bacterium or an exogenous virus. It could be just the normal production wear and tear of the lung. And in some areas, neutrophil, the epithelial cells and the macrophages that rely on CFTR to do scavenging through the endocytic role of CFTR. That is not present. And they basically that in that little area, the system calls upon professional scavengers from the blood to come and do their job. And this is what basically we're seeing. So there's slightly different hypotheses here at Emory and at UNC, but we all agree now to think that it's not triggered by infection.
Great. Thank you so much to our speakers and to the audience for those questions. I think we're just at time, so we'll probably finish up. Have a great day, everyone.
Thank you, everybody.
Great. Thank you.
Thank you, Rachel.
No problem.